Hello, my name is Aoife McLysaght and I'm from the genetics department in Trinity College Dublin, and it's a huge pleasure for me to talk to you today as part of your SciFest celebrations. And I'm sorry that it's not possible for us to be together in person, such as the way things are, but I know from previous years that the SciFest is such an amazing project and that the students, all of you, will have done extraordinary work to get to this stage, and I know the amount of work and the amount of dedication that goes into it, so I hope you're all feeling very, very proud of yourselves. So I work in evolutionary genetics, and I'm going to take this opportunity talking to you today to tell you just a little bit about the kind of work that we do and to give you a bit of an insight into what it is to study evolutionary genetics and why it's useful. And I think, and the first thing, one thing that you're going to see through this is that evolutionary genetics is not only useful for looking at the past and understanding the past, but also when we think about this and when we see how things have evolved, how genes have evolved to be the way they are today, it tells us a lot about how they actually work. And so it's a very important source of information for understanding present day genetics as well. So that's why I've titled the talk this way, that the past sheds light on the present. And I'm going to start with this very beautiful quote from this Dobzhansky, a very well-known evolutionary geneticist in his day, and he said that nothing in biology makes sense except in the light of evolution. And what he meant by that was we observe all of these strange things in biology. We observe strange patterns in terms of distributions of where animals live and where you have new animals or different animals that don't exist anywhere else, and where you find some animals that or other species that are present everywhere throughout the world. And we also see strange things in terms of leftovers or vestigial organs, like we have a vestigial tail, and we see things that are repurposed to a new function. So you have an older structure that gets a new function. And these things don't make sense except if you think about them in terms of how they evolved to be that way. So our vestigial tail makes sense because we are descended from animals that did have a tail, and so it's just a leftover bit. And the other repurposing of structures also makes sense when you think about it in terms of like an existing structure that was there that gets reused and repurposed. And we see the same thing happening at a genetic level. We see the same thing in terms of our DNAs. We see genes that are left over, they're just not doing anything anymore, and we see genes that have changed in what they do. So there's an old gene and it's changed and now it does something new. And when we look across the diversity of life on this planet, we see these endless forms most beautiful. And this is a phrase I've taken from Charles Darwin's famous Origin of Species book. And you can see just an example on this slide of some of the extraordinary diversity of life that exists on this planet. In fact, I'm only showing you animals here. There's way, way much more than just animals. But when you look at these and you think about them, you need to remember that all of these are different because they have subtly or dramatically different genes. So when we get these new functions and new structures and new forms and new colors, it's because the genes have changed. And so when we want to think about how this, as it has arisen in an evolutionary sense, we need to think about how this has evolved. And to do that, we want to think about, just remind ourselves for a moment, what is a gene? So we could have a long discussion if we wanted to, on defining a gene, because it's possible to think about it different ways. But one practical, useful, and everyday kind of definition of a gene is that it's a string of DNA that codes for a protein. So that protein could be the keratin in your nails and skin. It could be the hormone protein insulin, or it could be the globins that make up hemoglobin in your blood, carrying the oxygen around your body. All of these are proteins that are produced by genes. But when we look at a gene, I just want to give you a little sense for how genes can be complicated. So I've drawn this little horizontal line here, and this is just to represent a piece of DNA, just a stretch of DNA. I'm going to add little annotations to this to show you some of the things that we see when we're looking at a stretch of DNA that is a gene. So one of the things is we have of that big long DNA is made up of A, C, T's and G's, and of this we have the correct sequence of letters that maybe code for the protein that we're interested in here. So this yellowy orange bit here is a sequence of A, C, T's and G's in the right order that code for the protein we're interested in. But you'll see that but we also need some signal for where to start. So we've got a start signal or a go signal at the beginning of the gene, and also a signal of where to stop. And there's a stop at the end of the gene. But you'll notice there that I've got these orangey yellow blocks, and they're not all joined up together. So there's middle bits that need to be cut out. So we need signals for where they should be cut out. So this is supposed to be a scissors at the bottom, not exactly an expert drawing, but you get the idea. And then not only that, not all genes are turned on all the time. The different parts of your body are different, not because they contain different genes, they all contain the same genes, but because some genes are turned on and some genes are turned off at different places and also different times. So I have drastically simplified things here, but I give you, I hope, a picture of the fact that genes are quite complicated, and that tells you that evolving a new gene is quite a complicated thing, potentially. So genes are complicated, but DNA itself is simple. So here we have a photograph of a page of the human genome. So the human genome has been printed out in the Wellcome Trust Museum in London, and it's something like 60 volumes where the print on the page is really tiny, as you can see there in this picture. And this is just one page of the human genome. So it's extremely long, but it's simple in the structure. It only has those 4 components that we represent with the letters A, C, T, G. And you can see here you get the letters A, C, T, G written in their various sequence. So genes are complicated, but DNA is simple. And that means that it's actually simple to copy DNA, it's the simplicity of copying DNA is totally independent of the complexity of the genes. So when DNA gets copied all the time, so it gets copied from when one cell multiplies, so the DNA is copied and then the cell divides in 2. Now you've got 2 cells with exactly the same DNA, and it also happens every generation when the sperm and the egg or the pollen and the ovum are produced containing the DNA that's going to be the beginning of the next generation. So DNA can also be copied in another way. So you can get a copy, not a copy that gets sent to a new cell or when there's a division happening, but a copy that happens of just a segment of the DNA, and you get an extra piece within a cell. So duplicating genes is easy. So you have one gene and you can have 2 genes, and that's quite easy for it to happen, even though the gene itself is quite complicated. And this is a picture of an old telegraph machine, and it's just the same kind of idea. The telegraph technology was quite simple. It was just these little beeps that are being sent down the wire, even if the message that is being sent could be something really complicated, or it could be something really simple. It could be, meet me at the train at 5 o'clock, or it could be a very complicated message. It doesn't matter, the technology is the same. So the underlying DNA is simple, even if the genes it codes for are very complex. So if we say, where do new genes come from? We want to get a new gene. Well, most of the time, a lot of the time, new genes come from other genes, from old genes. So you get a new copy of a gene because it's just so easy to make a copy of the DNA, even if the gene itself is not so easy. So we see this as an important mechanism in evolution. We see a lot of evolution by gene duplication. And here's a little cartoon example of what I'm talking about here. You start out, you've got this piece of DNA that has within it the code for a gene. We're just calling it gene 1. And then we get an extra copy of this gene within the cell, within the DNA of the cell. And so now we just got 2 copies of this gene 1, so we'll call them A and B. And at the beginning, they'll be identical to each other, but over time, they are now independent pieces of DNA. So if a mutation happens here and it doesn't happen there, then this one becomes a little bit different. And again, another mutation might happen here in this copy B and not there. And again, we see that they become a little bit different. So over time, they become a little bit different from each other. And we see this a lot. We see this a lot in evolution. We have lots and lots of examples of duplicated genes in our own genome. So they change over time and become slightly different, but perhaps related. And the biggest example of this is our olfactory receptor genes. So the olfactory receptor genes are what give us our sense of smell, and they allow us to smell and recognize lots and lots of different aromas and odors. And we have about 900 of these in our genome. Over half of them are non-functional in us, but if you look at something like a dog has more olfactory receptor genes than us, and they're all functional. And that won't surprise anybody, I think, who has a dog or who knows about the extraordinary sense of smell that dogs have. So they have got this extra capacity in their genome to smell things. But this process of evolution by gene duplication has also been really important in terms of giving us our color vision. So here I've drawn this. This is supposed to be a picture of an eye, right? There's the lens at the front, and this pinky red bit here is a retina. So if we zoom a bit on this retina, and there are essentially 4 types of cells that I'm interested in there. So there are the rod cells, and then there are the cone cells, of which there are 3 kinds depending on which color opsin they express. So the opsins are the proteins that are produced from a gene that are light sensitive. And the rod cell contains the rhodopsin, which essentially gives you black and white vision. So that's just, that doesn't have color involved. It's just light and dark. And so that's what's in the rod cells. And the cone cells will express one of the 3 color opsins. So they only express one at a time. So they might have present in them the shortwave opsin. So that's the shortwave in terms of the wavelength of light, which roughly corresponds to the blue area of the rainbow. Or maybe the medium wave, which would be the green area, or the long wave, which is red. So we have these 4 different types of light receptor that's in our retina. They're all called opsin, so rhodopsin and then the other kinds of opsin, because they're all related by gene duplication. So there would have been originally one light sensitive protein produced from a gene, one gene producing a protein, and the gene got duplicated, and then little changes occurred in the copies so that that changed their light sensitivity so that they will react to different wavelengths of light. So when you look at these then and you think about it in terms of the spectrum of light, they have their maximum reaction at different wavelengths. And these combine then to give us our extraordinary color vision. So these 3 colors combine to allow us to see thousands of colors. So the different intensities of reaction at the different cones give us our color vision. Now that's in humans, but if we look at New World monkeys, so these are South American monkeys, we see something different. They don't have 3 kinds of color opsins, they have 2. So they have only 2 genes for color opsins. So they have rhodopsin like we have, and then they have the blue one like we have, but then they just have one other gene. So our red and green are on the X chromosome, and this is on the X chromosome in them as well. Instead, where we have 2 genes, they just have 1. So there's a gene duplication that we experienced that they didn't experience. So that one, there's one gene that's on the X chromosome in the New World monkeys. It can have variation in it the same way that other genes can have variation, and you know about variation giving rise to different eye color and different hair color and things like this. Well, there can be variation in this opsin, which means it's sensitive to different wavelengths of light. So here I'm showing you this particular individual has the rhodopsin, it has the blue, and it has the green, but another individual in the same population may, instead of the green, might have a version of the gene that's actually more sensitive to red light. So we can see these different versions of a similar gene or the same gene that exist. And so these monkeys then will only have 2 color vision instead of 3 colour vision, and so they won't see as many colours as we can. Another example of genes that originated by duplication, so where we get new stuff, very important new stuff in our genome, but where it's gene duplication that's been really important in that evolution, is in the globins. So you've probably heard already of hemoglobin, which is carrying oxygen in the red blood cells, and also myoglobin is carrying oxygen in muscle cells. So these are different versions of the same gene essentially. So these are related by gene duplication. So these new genes, different copies of the gene, came about because there was an original gene which got copied and then some changes accumulated. And these have changed sufficiently that they've specialized for different parts of the body, where the myoglobin is specialized for carrying oxygen in the muscle, and hemoglobin is specialized for carrying oxygen in your blood, which then of course transports it all over your body. And this is different localities in the body, but globins are very interesting because we have quite a number of them, and there's some others which are specialized for different times. So what do I mean by times? Well, developmental time. So if we look at this graph here, I've got this x-axis across the bottom, and this is showing the time since conception. So this is a fetus, an embryo, and then there's the birth, and then there's the age of the baby. And then the y-axis doesn't have a label, which is a terrible oversight, but it's just the proportion of these different globins. So these different coloured and labelled lines here are different kinds of globin. In an adult, as you probably know already, we have alpha and beta globin. So from about 2 years, from before 2 years onwards, this is what we have. But in the womb, there's different globins that are the ones that are important. So we see the alpha, this rises in frequency and becomes the main one for half of the hemoglobin early on. But instead of beta, which is low, we've got this lambda globin. And this is important because if you think about what has to happen in the womb and what has to happen at the placenta. So the mother's blood does not go into the baby or vice versa. They are kept separate, but they come up really close against each other in the placenta. So you have the blood vessels and from the fetal side right up against the blood of the maternal side. So the blood isn't mixing, but the oxygen has to get carried across this small membrane that's separating them. And this happens because the fetal or embryonic hemoglobin has a higher affinity for oxygen than does the adult one. So the mother will have alpha and beta globin making up her hemoglobin, but on the fetal side it'll be alpha and lambda, and because it has a higher affinity for oxygen, it'll drag the oxygen across that membrane, a bit like a strong magnet or something like that. So we see then just from these examples that gene duplication happens a lot, and it has contributed many important genes to our genome. But now we'd like to think about what we can learn from patterns of evolution, even within genes. And to do this, I'm going to take a little look at this story. And this is a story of a man called Abraham Wald who worked for the Air Force during World War II. And so the story goes that, so he was a statistician, and the Air Force asked for his help. They asked him to come and help them because they were trying to decide how to better reinforce their aircraft. And what they told him to do, so this is the way the story goes anyway, is what they told him to do is they asked him to look at the planes returning from battle and to look at where they were getting bullet holes. So if you imagine this drawing of a plane here, this is accumulated data over many, many different planes. So one plane had a bullet hole here, another there, and you put them all together and you collect all the data, and to look for patches where there were lots of bullet holes, and that would be where they would reinforce their planes, because they thought, well, these are the bits we need to reinforce so that we can get our planes to fly better. And you can't just reinforce the whole thing because metal is scarce and maybe it'll make the plane too heavy to fly anyway. And so the story goes that then Abraham Wald said to them, okay, I'll take the job, but you've got your logic totally backwards. I'm not interested in these places that have lots of bullet holes, apparently. I'm interested in these places where you have no bullet holes. So what's going on? What is he talking about? So let's imagine there's all these planes that are flying, and these bullets are firing up into the air somewhat randomly. So these planes get hit by bullets, and some of these planes then are shot down, so they don't return back to the base. And these ones return to the base, and these are the ones that Abraham Wald and the other people in the Air Force are able to see. So when they're looking at the bullet holes, they are looking at the bullet holes that don't stop the plane from flying. So these planes were shot, but they could fly anyway. So what you're interested in really is those parts of the plane where you don't see any bullet holes. And because you say, it's not that a bullet never hit in that part of the plane, it's that the plane couldn't fly afterwards, so it didn't come back. So these would be parts of the plane, like they were carrying the engine, or maybe the fuel tank, or maybe the steering equipment. And we can think about this same logic. This exact same logic applies to DNA and to genes. So mutations hit DNA randomly, and they make random changes in DNA, a bit like bullets firing up and hitting the plane randomly. And sometimes those mutations matter a lot, and they break essentially the protein, and other times they don't matter and the protein can still work anyway. And so what we see is, when we look at the way DNA sequences evolve and the way the DNA sequences, the genes evolve, we can see that some genes or some parts of genes really never seem to change at all. And that when you look at one in human and in mouse and in rabbit and in horse and in fish, you can see that the genes are almost the same across all of these different animals, or at least that part of the gene is the same. So that tells us that any time a change happened, it didn't return to base, a bit like those planes. So that tells us that's a really important part of the plane. And if we do that kind of analysis in terms of hemoglobin, we find that this center part of the hemoglobin really doesn't change much at all. It evolves very, very slowly. And this center part is the part that binds heme, which actually holds the oxygen. So it makes total sense. Hemoglobin is hemoglobin because it can carry oxygen. If you do anything to break that function, it doesn't work anymore. And whatever individual that mutation happened in isn't going to be healthy. And so that mutation is not going to be passed on. That's how natural selection works. So the pattern of evolution totally matches with what we know about the function of this particular gene. And then the same applies for genes that we don't yet know anything about. We can use this information, this evolutionary information, to understand what are the important bits and what are the parts of this gene that are really, really important to its function, and maybe then that can be our first steps in understanding that gene. So I started out with this quotation that nothing in biology makes sense except in the light of evolution. But I hope I've also convinced you that we can use evolution to make sense of biology. We can look at evolutionary patterns and understand then how genes work and where they have come from and what's going on. And thank you very much for your attention, and I hope you enjoyed that. And best of luck with everything else that you're doing for SciFest.